Okay, so welcome to PBS. Today's date is Tuesday, September 8th, 2020. Your warm up is in the discussions area and it's called 9820 warm up. And what you have to do is you have to respond to this prompt and reply to at least one other post. So the question is, what is DNA? How can DNA be used as evidence to solve a mysterious death? And does everyone have the same DNA? And you're going to respond to this prompt and reply to at least one other post. And then once you do that, that will serve as your warm up. After that, we moved on to a presentation. So I did the presentation today in Pear Deck, but that's okay. So pretty much the basic of it is what DNA. You're trying to deal with DNA evidence in this activity. So DNA stands for deoxyribonucleic acid, and it carries hereditary information of the cell. So that means that it's inherited from biological relatives. Every organism has DNA, and it's shaped as a twisted ladder, which is what you can see on the screen. And this is called a double helix because it has two sections to it. So how is DNA built? DNA is built from the monomer nucleotide. So the best way to think about a monomer is think about like a building block. So think about when you have those Lego pieces. Each Lego piece is considered a monomer, and those multiple monomers build something great. So that's the same way that DNA is built. So it has a nitrogenous base, which are A, T, C, and G. So those are the different bases. A is for adenine, T is for thymine, C cytosine, G guanine. It also has one sugar, which is equal deoxyribose. So anytime you see an ending in OSE like fructose, glucose, sucrose, those are sugars. And it also has one phosphate. So which bases pair together? So C is always paired with G. So cytosine and guanine always go together. And A and T, adenine and thymine always go together. And these are called Chargaff's rules. So the amount of cytosine equals guanine. The amount of thymine equals adenine. So C always equals G, T always equals A. And the total of all four is 100%. So they are so how are the bases attached? Like what keeps the bases together? Since I said like they're like puzzle pieces. So you have something called hydrogen bonds, which are weak bonds, but they hold the bases together. So that is how they're able to stay together until they're needed to separate for DNA replication. So remember we talked about Chargaff's rules and I said A equals T and G equals C. So if I have 30% adenine like for a human, then I know that A equals T. So whatever A is, T must also equals the same amount. So if A is equal to 30, T is also equal to 30. So when you add 30 and 30 together, you get 60%. But remember, it has to add up to 100. So when you take 60 away from 100, you're left with 40. So you still have guanine and cytosine. So whatever G is equal to, C also must be equal to. So if you take that 40% and divide it by 2, you'll get 20 and 20, which you can see in this chart. So if you find one piece, whether it's G or if you find A, then you're able to fill in the rest of the chart. So DNA looks different at different times. For example, when the cell is not dividing, DNA looks like a tangled hairball called chromatin. It looks like just a whole bunch of like hair all together just matted up and tangled up. So think about like when you take a shower and you wash your hair, like sometimes it's just like one large big hairball. That's what chromatin is. But when the cell is dividing, like mitosis and meiosis, DNA condenses and is organized into an X shape called a chromosomes. So how many chromosomes do humans have? So just think about that and we'll get back to that question. So again, DNA coils into chromosomes when the cells are dividing. So humans have 46 chromosomes. You get 23 from mom and 23 from dad. Or it can also be called as 23 pairs. So think about a pair of shoes. You don't just have one, you have two. So 23 pairs will also equal 46. So you have half of your DNA comes from your mother, half of your DNA comes from your dad. If you look at this chart, you'll notice some things. You'll see that different organisms, different species, different things, they have different sets of chromosomes. If you look at a human and you look at an orangutan, you'll see that we're more related as far as DNA and chromosomes than we are to like a frog or a garden pea. So a lot of times smaller things, they have less chromosomes, and things that are much larger, they have more. Or unless certain plants also have more chromosomes because they have to do more things than we do. So again, this is just a picture of the DNA structure. You have those bases in the middle. On the outskirts, you have those phosphates, which look like little satellite dishes, and the little houses, those are the sugars. So just some practice, match the bases below with their complementary bases. So you know A goes with T, so you write T at the bottom of it. T goes with A, and you just keep going. What is the name of the bond that connects the complementary bases? Where in the cell can DNA be found in a eukaryotic cell? And where in the cell can DNA be found in a prokaryotic cell? So those are just some things to like think about. So then going into the lesson, so you're going to go to assignments, and you're going to go to activity 115, DNA evidence. So in this lesson, you will take notes in your lab journal on the structure of DNA based off of the lesson and teacher video. We're not going to do the 3D model of DNA or anything like that, and we're not going to do an extraction because we're working virtually. So the goal is to explore the structure and properties of DNA, examine how and why DNA is used in forensic science, discuss what tests are used to analyze information in DNA. So to access this, you will click on this link, and then this will pop up. And I'm just going to scroll to the top. But it would be activity 115, DNA evidence. And we're doing distance learning, so we're not doing any of the physical part of it. So the first step is the DNA debrief. You're going to title a new page in your notebook, 115 DNA evidence. Read the entire section, take notes as you go, and you're going to make a video using this information. So be sure to understand what you are writing. So you don't have to start the video right now with making it because you have to add another piece to that video. So just make sure as you're taking those notes, you're knowing what you're writing and what you're going to put in the video. And it's just pretty much an information type of piece. Then you're going to move on from the DNA debrief and go into the DNA structure, where again you're reading and taking notes, getting information. And what you have to do in this section is you're going to read the entire section, take notes as you go, and you'll make a video using this information. So you're going to add this information with this one, and you're going to have that as your video. Then what you have to do is, since we're not physically doing it, you're just going to go straight from taking the notes to click here, check for understanding. And it's 4 questions you have to answer. You're going to take a screenshot of your results. So if you keep scrolling down here, you're going to complete the check for understanding after step 1. Take a screenshot of your results and paste them into a Google Doc titled 115 DNA evidence. So you're going to start up a new Google Doc like how we always do with our notes. And you're going to title it 1.1.5 DNA evidence. And you're going to post screenshot. So then now you're done with that part. Then you're going to go to your unique set of instructions. So after you're done with that, you go down to the next section, which is your unique set of instructions. You're going to read this information and you're going to take notes and answer the prompt in your notebook. And then that's all you're doing for that section. Then after that, step 6 is finding DNA inside of cells. You're going to take notes as you read and follow the directions for steps 2 and 3. But if you look at it, step 3 is actually not there. So you're really just answering for step 2, and you're going to fill out this information. And that's all you're doing for that. So most of this activity really is, because we're not physically doing the extraction and taking the DNA, is we're just doing a lot of reading and writing. So then you're going to move on to DNA profiling, which is here. And again, you're just going to read that section. So you don't even have to take notes on that section. You're just going to read this section and understand what DNA profiling is and what that looks like. Step 8 is the sources of DNA and extraction. So this is where we're not going to be able to do that lab because we're virtual. So you're just going to, again, take notes. So you have some flip cards. So you have 6 flip cards. You're going to click over all of these. And you're going to take notes on all of these different things. So like for the first one, you have red blood cells. You have bone. You have hair, preserved body parts, clothing and fibers, saliva/body fluids. You're going to take notes on all of those. And you also have a reflection question. So you're going to flip the cards and take notes, respond to the reflection question. So the reflection question is, what are some possible sources of DNA from the scene where Ana Garcia's body was found, and explain. So all of this is going into your notes. Then you're going to write down the 4 basic steps in DNA extraction, which is here. So these are the 4 basic steps. 1, break open the cell and nucleus. 2, purify the DNA of any cell proteins. And you're just going to keep going. Then after that, you're just going to skip all the way down, because again, we're not doing that. So you're going to see where you see the distance learning support. You're just going to click that and go straight to that reflection question. And that's what you're going to answer. So what are some challenges in extracting DNA from samples related to an active investigation? So that's what you're going to answer. And then you're done with step 8 of your Canvas part. Then going down to step 9 is prepping DNA evidence for analysis. So you're going to take notes again in your notebook as you read the section. So you're just going to take notes, read the section, and just take notes on that. You're going to skip step 4 because you're just taking notes for this section. And then step 10 of your Canvas is you're going to title a section of your lab journal 115 conclusion questions and respond to the questions in complete sentences. So you have 2 conclusion questions. And these are the 2 that you're going to answer. So DNA has 2 strands. If the sequence of nucleotides of one strand was known, is it possible to understand the information to determine the sequence of the second strand? Explain your reasoning for response using an example of DNA sequence. 2, can you be framed by your own DNA? How might an individual's own cells lead to false verdicts of guilt? In your explanation, provide one scenario in which this might occur. So you're going to answer that. Those are your conclusion questions. What you're turning in is the link to your video. So remember, in steps 2 and 3, you had to create a video. So you're going to turn in that link, screenshot for your check for knowledge results. And we're not worried about this because we're not doing the cheek cell DNA. And then just picture of your lab notebook for the week. So if you look here, it explains to you how you're getting marked. So don't worry about the origami part because we didn't do that. Just worry about the video part and the lab notebook and a screenshot of the check for understanding, because again, we didn't do this part. So you automatically get the points for the origami stuff and the student design nucleotides. You automatically get the points for that. So what you have to worry about is the video, the lab notebook, and the check for understanding. And then that's pretty much it. So you should be able to finish all of this by today, as well as I did explain that you will be getting progress reports this week. So you do have to make sure all of your other previous activities are in, including 114. So I did extend that because I know some people weren't able to finish it over the weekend. So just make sure that you turn that in as soon as possible so that you can have the most updated grade, so that way your progress report will be a true reflection of what you've done thus far in this class. Any other questions, you can just email me or message me on Remind so that we can make sure that you have as much success as possible. And that's it.